Okay, so today we're going to discuss this paper, Transforming Insect Population Control with Precision-Guided Sterile Males with Demonstration in Flies. So this is the pgSIT, Precision-Guided Sterile Insect Technique. Okay, let's just look at, so this is a paper from Omar Akbari's lab. Akbari is now UC San Diego, I think. And he's a very famous mosquito geneticist. So it's worth discussing this paper. So here's the scheme, which I described for you yesterday or the last time for the pgSIT. So essentially the gist is you make a homozygous line for Cas9, that's this line 1, and you have a second line. And here's where it actually gets interesting. So I skipped over some of the details last time I talked about this, and as reading the paper, I learned a lot more about it. So in the second line, it's homozygous for two guide RNAs. There's a blue one and a green one. Did you catch, well, it explains it in the diagram, but what are these guide RNAs? What do they do?
They either cause sterility in males or a lethal factor.
So sterility in males, and then here's lethal for females. So you get like a two birds, one stone, 2 in 1. Which translates to sex sorting and sterility all in one step. And if you actually read their introduction pretty closely, that's kind of one of the main benefits that they're arguing for this system is you get simultaneous sex sorting and sterility. So everybody, this makes sense. There's another key thing that I did not catch until I read this paper in detail. Did you catch in the other sterile insect techniques that we have discussed, what are they releasing? Like, what stage?
Adults.
Yeah, they're releasing adults. What are they proposing to release here?
Sterile males.
No, yeah, eggs. So this is unique from the other systems in that they're not actually proposing to release sterile males. They're going to do these crosses, and then they would get eggs, and then they could release egg rafts. So it's kind of weird to talk about this with the diagram of Drosophila because they do the proof of principle in Drosophila. But with a mosquito, you could get them to just lay the eggs from these crosses on like a raft, or I mean, not a raft, but like a paper. And then they would just like, I guess, distribute the papers as a means of distributing the...
Do you think that could be like a secondary competition?
What do you mean, secondary competition?
So you have sexual competition with sterile males with fertile females, but also you can have competition by releasing a ton of eggs.
Yeah, they talked about that. They said they kind of just talked about that as like another pro of this technique is that because you're releasing eggs, these larvae are going to be competing with the local populations as larval stages too, not just for mating. So they kind of say that that's like a plus. Okay, so that's essentially like how it works before we go into any of the more complicated data. So let's also just kind of like write out why do this? Why do this, do these experiments and do this research? What are your thoughts?
Less labor.
It's interesting. What part of it would be less labor?
They don't have to get a machine to sex sort.
Okay, so less labor in sex sorting. Yes, that's a good one.
100% efficiency.
It was 100% efficient in Drosophila in the cages. But that's what their data showed. That was nice. But that's not why you would do it. Why else would you do this?
In the instance of eggs, it's super easy to store them. So I would think that'd be really useful for release efforts.
Oh, that's an interesting thought. I did not think of that. So the fact that you can store eggs.
So if you have an area that's really bad.
Yeah. You might be able to stockpile them.
Yeah, and then you could basically mitigate how much you release in an area based on
Yeah.
how bad it is.
Yeah, the one thing, so that's a really good point. I did not think of that. Oh, yeah, okay, so I was wondering if a case with releasing eggs versus releasing adults. One thing that I was wondering about is if you are releasing eggs, would you get adjust this so people can see my face, like a patchiness effect? You know what I mean? Like imagine you had like egg papers and you were like walking around and you put this egg paper in this pond, this egg paper in that pond. Would you get enough distribution of the males that way?
Possibly because they can travel mosquitoes on a daily basis can travel up to like a mile in radius.
Maybe. I know that in theory like some can travel a mile. In some of the papers that I've seen, they don't like cross roads and stuff.
I think in the release effort it's going to be more difficult because you're not releasing like that mobile phase. It's going to be like a problem. But
Yeah, see, that's exactly what I was wondering about. And I don't know. Like, anyway, I'm not doubting it. I'm just curious to see if the release of the eggs is as evenly distributed as like releases of adults. Maybe it's better. I don't know. Why else? Okay, so why else would you do this? These are all good, and you're getting things that I did not think of, so this is good. Anything else?
With the regular sterile insect technique, is there a loss in fitness for the males?
Depending on, so that's another reason is sterility without loss of fitness. There isn't a loss of fitness with Wolbachia, and I don't think there's a significant loss of fitness with the riddle, but there is with the radiation. So I think this is just like one of the other tools we have now that is not giving them a loss of fitness, but is giving them sterility. Why else? Why use CRISPR-Cas9? Like there's actually a point to using why you might want to use that. There's a bunch of reasons why you might not, but there's also reasons why you would want to use that. It is precise, yes, but you can do a lot of different other precise things.
It's relatively cheap.
I think all sterile insect technique is actually going to be really expensive.
They want to maintain the male fitness too?
Yes, they are equally fit. What else? Essentially, okay, so they do it in Drosophila, but yet they're talking about it being applied in mosquitoes. Why does that make any sense?
It's applicable across like species.
Yes, that's the thing. So it's like cross-species compatible. Because CRISPR-Cas9 tends to work in essentially like everything. So that is one reason why you might go out of your way to develop this. Whereas if you're developing some very specific sterilization mechanism of some weird genetic system in some weird insect, it's only going to apply to that insect. Whereas if you build it off this fundamental toolkit, it should be able to be essentially like applied across other insects. You might need to change a few things. Like what things would you need to change? Yeah, you'd probably need to test a new array of like guide RNAs, a battery of guides to see what works best. But essentially like this system, the logistics of it is going to be essentially exactly the same, depending on what any insect that you're working with. Yeah.
So I didn't know anything about CRISPR-Cas9 before this class really. How applicable is this to humans?
Like, could you do this in humans?
Well, maybe not like this experiment per se, but like what is the results of CRISPR-Cas9 in humans?
Well, they've gene edited babies. I mean, that's already been done.
Oh, really?
Ethically,
Yes.
Damn.
They did that in China, and I think the professor is now like not a
I
professor.
was going to say, so is that considered eugenics?
Well, I don't know what you would. Oh God, this is gonna. I don't know what you'd consider eugenics. I mean, the ethics of that were questioned because I don't think they did a thorough investigation of off-target effects in those human genomes. So the worry in that case was are you creating these babies that have some other defect that you don't know about? I don't know if they sequence their genomes or not. Although I think it's going to become more and more common. I don't think it's going to become less common, if that makes
I
sense.
feel like the concept of designer babies really rubs some people the
Wrong way. Yeah, I don't think it's going to go to that yet. It's not going to be like, oh, choose your eyes. Like that's what they always kind of like say. In that case, they were trying to. The justification for that was they were trying to take a father who had AIDS and make sure that he can have a baby without AIDS. So they did the fertilizations, and then they CRISPR modified the babies, the embryos, to essentially have an HIV resistance gene.
Inside the
That was in the chest tube, and then they implant the embryos in the mom. Does that make sense?
Yeah, that's type.
Yeah. So, and I think that implementation was also somewhat questionable. Like I don't know if I don't know. That was a justification though.
CRISPR for like gene therapy, like gene replacement. Do they use something else?
Well, for sure, I know there's research in that.
Because I know that there's like some loose, like, I don't think anything's on the market, but like clinical type application.
Yeah, so there's going to be a lot of clinical applications that are going to be trying to utilize it to essentially change somatic cells. So like if you had cancerous somatic cells, if you could re-edit their genome to be non-cancerous or something like that, I don't know, that would be useful.
Use CRISPR for that.
Yeah, but it's way more controversial to do it in germline tissues that are modifying essentially every cell of the organism so that then when that baby grows up and has more babies, they will inherit that.
Oh, that's scary.
And yeah, so probably it would eventually be some kind of like eugenics regime. Anyway, I think it's going to become more common, not less common. Anyway, okay, that's a CRISPR tangent. Anyway, like these are reasons why you would do this. So we all understand like why we would do this. Let me also just quickly, like, I think this would be a good thing to have on the test. Like, let's just review pros and cons of the various techniques that we've discussed for sterile insect techniques. So there's the riddle, there's the IIT, and then now there's the pgSIT. Okay, and they kind of go through in the introduction here, and they kind of slam some of these other methods. Did you guys catch what their cons were? So in IIT, what's the con of IIT? I think they're correct in some sense about this.
Wolbachia being sort of organism specific.
No,
But you can't release any females.
you can't really. The danger here is the risk of releasing females. They write in the text something like, not a single female can be released. And I think I think that's actually wrong, because in order for Wolbachia to actually initiate like a gene drive, it has to be over a threshold. So a release of one female is not going to do that. But the people who are doing the Wolbachia releases argue essentially like it is a risk, but you don't have to worry about like a single one. It's going to be like a few, more than one. But it's a fair point. I do think that is the biggest con of the incompatible insect technique. They're right about that. What's their con for riddle?
The antibiotics destroys the microbiome.
Yes. Good. You read this. You read this very well. Good. Antibiotics potentially reduce the fitness because it's messing with microbiota. And I think that's a fair point. Although the response to these are, guess what? Like riddle's been used and it seems to work. Guess what? IIT's been used and it seems to work. Although theirs could work better, you don't know until they try it. So I think it needs to be tested. It needs to be tried in the field, the precision-guided sterile insect technique. Let me just go through my notes here and make sure I get everything. Remember here, so for the pros, so if we're talking about like pros and cons, the pros of IIT is it's already in use and it's non-GMO, okay? A con of riddle is that it's GMO, but a pro is that it's already in use and it already has like a regulatory framework for approval, okay? The cons of this system would be, I think that it's GMO and there's no usage case yet. In terms of like, has it been applied yet? And is there proof that it works yet? I don't think, although I could be not up to date because this paper is, I think, a 2019. I think they're gearing up to do some field tests. So it would be interesting to see if this works. And if that's wrong, then forgive me. Let's see. I do think that the pgSIT, okay, so let's list their pros. Their pros are you can release the eggs. That's a good one. The pros are that you can do simultaneous sex sorting, and sterility. And I think that's a big deal. I don't think that's like, I'm not, that's a big deal, the fact that you could do this. There's one final con to this, which we talked about last time. Do you remember what it was? How many lines do you have to rear? Yeah, you got to rear 2 lines. And that also means if you're going to do these crosses, you have to collect virgins. Okay, like researchers can collect virgins in the lab. I don't know how they're going to scale up collection of virgins. I don't know if they'd have some kind of like machine learning contraption where it sex sorts and then sets up the crosses. I don't know how they're going to solve that. But they have to somehow solve how you get virgins and how you set up the crosses. And I think that might be a little bit difficult on a scalable size. So,
Like the bulk of their work comes from.
what?
That sounds like that would be like the bulk of their work in lab would just be.
Oh, like it's interesting. When I talk to people who are doing transgenic research on mosquitoes, that's one of the first things I ask them is how many lines do you have to rear? Like the graduate students, how many mosquito lines do you have to rear? And I've talked to people and they're like, oh, I rear like 20 lines. And yes, they're like 50% of their workday is like maintaining transgenic lines. It's no joke. And I would not imagine how hard it would be to do something like this on a scalable level. Although, like, I do think there's room for machines and computers and things like that. Essentially, like, the point is, okay, all these technologies have pros and cons. And I don't think it's going to be one that sort of like is the best bullet for all the scenarios, if that makes sense. It's always going to be weighing the options kind of thing. Okay, so what's the first thing that they set up? What's the first like test that they did? Actually, we can quickly talk about this. This figure kind of made me scared a little bit. How do we interpret this figure? Although it's not too bad.
Just the gene structure of the alternative spliced genes.
Yes. So remember back to our sex determination. In Drosophila, and it's somewhat similar in mosquitoes, but different. The alternative splicing in Drosophila regulates sex differentiation. And so they have these 3 genes. They're just pointing out sex lethal, Tra, and double sex. These are key genes in the sex determination pathway. And essentially they just pitched this as, well, if we're going to try to kill females or convert them, like remember the Nix paper, like we're going to make some hermaphrodite flies, we'll target these genes with guide RNA. We'll do some knockouts and we'll kind of like see what happens. That's essentially like the gist of this figure. So don't get too wrapped up in trying to understand like what the alternative splice transcripts looked like. Just understand that's what they're pointing out here. Okay. The next figure is actually data from experiment 1, their first experiment. So their first experiment is sort of just testing to see if the framework works. The sterility and the sex sorting. Okay, and so this like panel here. So first, let's just split this in half. Like, don't even worry about this side, just worry about the left side. So what's this first panel? What are they showing you? What is this? It is a control. And so because it's a control, what would you expect to observe and what do they observe?
Really no changes in ratios or sterility.
Yes. So sex ratios in Drosophila, if they're normal, should just be 50-50. And so here's your 50-50 line. And essentially they're seeing about 50-50 sex ratios when they do these particular crosses. This symbol, that's like the hermaphrodite symbol.
Hermaphrodite Drosophila common within like crosses.
Well, wait, say that again? No, it's not. Because this would, if it was common, that's an interesting question. If it was common, this would be higher, right? So no, that's like completely rare. So it is interesting that they even look for it. But they're looking for it as a negative control.
They see it in the actual like.
Yes, because that's what they're trying to do. Okay, so now down here, now is where they're actually testing like their constructs. They're testing their weird constructs. Okay, so here they have 1, 2, 3, 4 different guides. So essentially in the article, they say something like in the introduction, we had these like 9 genes that we were interested in. Some of them were sex differentiation. Some of them were important for sperm motility and things like that. And they're trying to figure out which of these are going to be useful. Okay, so this one is guide RNA that's targeting sex lethal. And what happens?
They all become fertile males. Or all the offspring that is just, yeah.
So essentially what's happening is they don't all, quote unquote, become fertile males. It's killing the, is it? My interpretation was that it was killing the females.
It's killing the females, but because that 100% is just the only leftover population.
Yeah.
Ratio.
So I think my interpretation of this is killing the females. I don't think it's actually making fertile hermaphroditic ones. Okay, so what's the interpretation of this cross? Here's where it starts to get interesting.
It starts creating like hermaphroditic Drosophila.
Yes, so the blue is the normal like fertile males that are just already there. When this guide RNA gets hit or hits the transformer gene, it's taking females and it's actually converting them into the hermaphroditic males, and they're sterile. That's this like orange thing. So both this one and this one are giving that same effect, transformer and double sex. Okay, and then what's this last one? And how is this one a little bit different from the others?
The males are sterile.
Yes, this one is essentially, it's only doing something to the males. And if you look closely, this is beta tubulin. This is a sperm-specific beta tubulin. So it's essentially like an essential gene that is important for sperm function. And if you knock that out, you just get sterile males. So that's essentially like the gist. It's kind of a intimidating figure to look at, but it's really pretty simple, and that's what they're doing. Okay, so that's the left side. What's the right side, and why does it matter?
In this one, the males are edited. They're being mated to wild type males.
Yeah, so it's essentially like the reciprocal. It's just the same experiment with the reciprocal cross. Why would that matter? Like, why would you want to test that?
To see if there's a difference.
Yeah, it's as simple as that. Like, they're trying to set up a system here where they're going to try to scale it up eventually and convert it to mosquitoes and scale it up. And so they just want to know, like, do they observe any difference? Is it important how you set up the crosses, etc.? And they observe essentially the exact same phenotype. So what are the conclusions from these data?
The crossing into wild type flies has no change in morphology or sex ratios.
Say it again.
Are you talking about the control?
No, not the control. Like comparing the data against the control. What are your conclusions?
I mean, it just gives you these different options. It depends on whether you're wanting to kill females or sterilize males.
Exactly. So that's a good point. Is first of all, it works. And I think Alex said something like it works with almost like 100%. Excuse me. 100% efficiency, which is pretty amazing. And as Rachel suggested, you actually have 2 options. One option is essentially like convert females into the hermaphroditic males that are sterile. And the other option is kill females and then sterilize males. So you have like a couple different options here. This also includes make sterile males. So it's essentially, you can start to now decide like which of these is going to be best. I don't know what's going to be best. Maybe they'll do some more experiments later to figure that out. Okay. Test 2. Here's kind of the big figure from experiment 2. What are they doing in experiment 2? It looks very similar, but how is it different?
They're doing a cross with the like both gRNAs, both guide RNAs.
So they're doing a few different things. So they're combining, it's like a combinatorial experiment. And you're saying they're combining different guide RNAs. What else are they combining or testing?
It looks like similar to the last experiment, they're seeing like which cross works best.
How come the last experiment only had 2 and this one has, 3 and 4?
They're using different promoters.
They're testing different promoters. So their idea is we have all these different combinatorial guide RNAs, and we could combine them differently with different promoters for Cas9. Okay, so let's quick just look at these, and let's describe them. And then we will also look at the guide RNAs and describe them. So if I zoom in here, nose. So I'm not expecting you guys to know advanced insect genetics, but it's worth knowing what these things are. So I won't put like nos on the test, like what is nos, but I will put potentially something related to germline promoters. So nos comes from nanos. Nanos is a developmental gene. So it's a germline promoter. Vas is very similar. Vas comes from Vasa, the Vasa gene. That's very similar. These are two germline promoters. And these are, if you are interested in insect genetics or genetics in general, these promoters come up all the time in scientific literature. What is this promoter? This promoter also comes up all the time in scientific literature. Ubiquitin. Ubiquitin is essentially like one of the most commonly expressed proteins in all cells. And so it's a housekeeping gene that you need. And so its promoter is very strong. So it's a strong constitutive promoter. So ubiquitin is always tested as like a strong constitutive promoter, which means it's always on. These ones are tested as strong germline promoters, which are on typically in the testes, ovaries, and early zygote. Okay, now let's look at these guide RNAs, and let's make sure that everybody understands how to interpret these guide RNAs. Okay, so when you see the gRNA, that always means guide RNA, and then they have the superscript here, beta tub. What's beta tub? Tubulin.
Like a sperm protein?
Is tubulin a sperm protein? Yeah, tubulin is kind of like a essential cytoskeletal gene. You need it in all cells, otherwise you die. But sperm have special sperm-specific tubulins, and I'm pretty sure this is a sperm-specific beta tubulin. SXL, that's sex lethal. So this guide RNA is essentially like double guide RNA. I think that's what DG is, double. Somebody check me on that. I think it's double guide RNA. So it's two targeting beta tubulin and sex lethal, or beta tubulin and TRA, beta tubulin and double sex. So they're testing the simultaneous sterility plus sex sorting. Okay. And let's see. So in this one, what's the result?
All sterile males.
All sterile males, and this is a female killing. So it's killing the females. So sex lethal, if you knock it out, it's killing the females. And these two are then the other option where you get full sterility of the wild type males and then conversion of the females into sterile hermaphroditic males. Does that make sense? So there's kind of like two again, these are like the two options that they're creating that you might want to pick. And then the left side, if they just do the same thing with the reciprocal crosses on the right side. So it's pretty easy to interpret the data.
Dr. Beckmann,
Yes.
how would you interpret it if, like, on the other side, the results were just completely different?
Well, that could happen for a few different reasons, right? That's a good question. Like, even though they're the same, like what conditions could create a situation where they would be different? They do kind of talk about some situations that might create that in the text. Did anybody catch that? So if it comes from the female, what's typically happening in transgenes that are expressed in the female that get put into embryos? They're typically they can cause maternal effects. And so there's a scenario under which perhaps, so imagine you put Cas9 in the female compared to a Cas9 in the male. And let's say it works with like, if you have robust Cas9. In the female, you're going to get really, really good loading of Cas9 into the embryo because of what are called the nurse cells. I kind of drew this out once before, didn't I? So if you have an egg in essentially like developing embryos, it's surrounded by the germline-derived nurse cells. And the job of the nurse cells is to literally, all they do is they take nutrients and they create messenger RNAs and proteins and load them into the egg for the egg to use for development. And so if you have a female expressed Cas9, that's germline, it's going to get pumped into the egg from the nurse cells. Does that make sense? So you could envision a scenario where you get super robust Cas9. Contrast that to what's happening if you express Cas9 in the male. If you express Cas9 in the male, okay, it's got to essentially end up in a sperm cell. The problem with delivering things in sperm is that as sperm is developing, transcription essentially like shuts down. So as sperm cells are developing from stem cells to adults, there's a point at which the chromatin has to become compact. Does that make sense? To like fit and protect itself inside of the sperm. And as soon as you compact chromatin, guess what happens to transcription? It stops. So essentially like 95%, maybe 95%, maybe 99% of all transcription in sperm cells like shuts down. So if you try to express transgenic Cas9 in a sperm cell, it might not work very well. It might not give you very high abundance. And so there are scenarios under which you could deliver a transgene and then as soon as it hits the zygote, maybe it would turn on. Or maybe you could figure out some mechanism of like loading it in the sperm. But essentially, this is one mechanism through which you might expect to see some kind of differential ability for this to work. Does that make sense? But they did not observe that. They observed that it worked totally fine. And it probably worked totally fine because I think they talk about they're getting some zygotic expression of the Cas9, which is totally sufficient. Does that make sense? If it's delivered from the male. That was a pretty complicated discussion. No questions on that? Okay, good. I'll put that on the test. Okay. And then they do kind of like a third experiment that I don't really want to talk about, but here's some interesting things. What's the gist of like these figures?
They're like visualizing the phenotypes of these sterile bugs.
Yeah, they're visualizing the phenotypes of these hermaphrodites. So just like the next paper, these are essentially, so Drosophila have what are called sex combs. That's these things. If you don't know what to look for, these things. I'll get a better one, get a red marker. These things are sex combs, and the males will like pet the females with their sex combs. And so here are females that are developing sex combs, which are pretty interesting. Here's wild type ovaries. And then I think in here there's some suggestion that like the hermaphrodites are like developing. Here's a hermaphroditic ovary where like one formed. Essentially like they're showing you the hermaphroditic phenotypes. I could pay like one, if I really was obsessed, like I would pay more attention to like, okay, which guides are creating certain ones. And I think they do care about that a lot. I essentially saw this data and I was like, okay, it works. Yeah. Okay. Then what's their sort of like, what's a final thing that they need to do? What's this all about? This is sort of, I think this is something like experiment 4. Assess the fitness. I thought it was actually interesting in a sense of, this is not how I would envision you would measure fitness of Drosophila. I've seen Drosophila fitness measured in like crawl speed and things like that. Like you can do weird experiments where they like crawl up the side of the tube and you can measure the speed at which they like crawl up the tube. I've seen people analyze like mating dances. They're kind of just doing crosses. And then what they're measuring is a readout of hatch rate. And I think it works. Like it works for what they want to know. They essentially just want to know, are the matings happening at a relatively reasonable proportion? And I think this is fair for that. Although I don't necessarily think it's a good measure of sort of everything that encompasses fitness, if that makes sense. So within this figure, this was also kind of like an intimidating figure for me. How do you interpret this figure? What should you, walk me through how to understand this.
They're just showing the different crosses.
Yes, okay, that's the first thing. The first way to look at this is first understand that they're doing 4 different crosses, and they're comparing 4 different crosses. Okay, so the data over here come from these crosses. How do they set up their crosses?
It's like,
Yeah. Yeah, so okay, so this is a negative control where they're putting one of these wild-type males with 10 virgin females. Here is another negative control where they're putting 2 wild-type males with 10 virgin females. And then cross 3 is where sort of like the action starts happening. This one's really important because here they're doing, this is actually a competition. It's a competition between the transgenic male that's sterile and the wild-type male that's fertile, okay? And then this one is the double sterile, okay? So red means sterile. Okay, and these data are a readout of that. When you're gonna look at this, first just ignore the laid eggs, ignore the white bars, because it sort of just makes it harder to read. Just look at the hatched eggs. So essentially, hatched eggs is hatch rate. So this is a 0% hatch rate, just kind of like CI data. And these are the 4 crosses. Okay, wild type, this is the wild type. They're getting a hatch rate of about, this is probably like 85 to 90%. That's wild type, that's normal. Ditto, this is not significantly different in the second cross where they also have 2 wild type males. And then here's where you see the action. So in the competition assay, the hatch rate is exactly at 50%, essentially, the means. What does that tell you?
The sterile male has the same chances to mate as the normal.
The female cannot tell the difference at all. If she was able to tell the difference, you would have some. It would not be at 50%. It would either be high or it would be low. If it was low, she's preferring the sterile male. If it was high, she's preferring the fertile male.
But
So
you can conclude, like, based on that the sterile one is better, right? Because it's just half.
well, no, I'm not saying it's better. I agree with you. I'm saying she can't tell. She can't tell a difference. That's what I would conclude. Is that fair? Yeah. What I was saying was if this mean was up here, then you would conclude that she could tell a difference, I think.
Well, that's all. So my question is, like they only mate once though? Like she can't mate with another male after she's mated with one of them?
In Drosophila, they're only going to mate once. There's no okay, fact check me, but there's no, this experiment implies there's no sperm competition. Yeah. Yes. And then this one, her only option is sterile males, and they're all sterile. Okay? Any questions? That's how I would interpret that. Okay. And then they do this, essentially like this model. And like it's kind of like, okay, like our method is the best, which I think it makes sense. Which I think makes sense, like in a sense of like that's great for them. They definitely like want to show this. So they do this model, and it's essentially how many things do you have to release to get good suppression of the population, and how fast is it? And it's like a computer-generated model where if you're releasing, they say that in IIT studies, they're releasing at 10 to 1 adults. In normal riddle, they're releasing at 10 to 1. There's this other weird version of riddle where they're releasing eggs, and they're releasing 200 to 1 eggs, and then they compare their pgSIT to that. And their pgSIT is essentially like crashing the population right here at this blue line first, before any of the other methods. So essentially like their conclusion is, yes, this is going to be real good. Now we are ready to transfer this into mosquitoes and do some field studies, perhaps. Any final thoughts?
The one thing that is kind of like interesting is the ratios on which they have to release to cause that crash.
Yeah, when I was looking at this, it took me a second to interpret this, and then that's when I discovered, oh, like they're planning on releasing eggs. Yeah, okay, that's that, and so that is interesting to me about the pgSIT.
That's probably
Why? This is an interesting question. Why can't you release eggs with IIT?
You have to radiate.
What do you mean?
Well,
Why do you have to release adults with IIT? Explain that to me. So say you get a batch of eggs and you release them. Why is that bad for IIT?
You can't have females.
Yeah, because 50% of those eggs are going to be females. So you cannot do release eggs with female or with IIT. Okay, tell me the same question for Riddle. Why can you not, for classical Riddle, why can you not release eggs?
because they need anabolic treatment during the normal stage.
Yes, to stay alive. So, yeah. So you can't, that's the other reason you can't release eggs with the classical Riddle, although there's a newer version of Riddle where you can do these egg releases. And so I do think that's a really interesting thing about this technique, that it's pushing the technological things that we can do in a sense of with sterile insect technique, it's allowing you to do these egg releases. And I'd be really curious to see how easy or perhaps harder that is. I don't know.
I was also pointing out the huge disparity in the different ratios, which makes sense because since they're releasing egg stages, not all of them are going to like emerge as adults after a while.
Yeah, I think that's why they're doing that.
It would be cool though if they had matched the ratio to see what it would be.
1-to-1. Well, I think, yeah. I don't know.
If they matched the ratio with like IIT and Riddle, and it still showed it, that would also, it could promote like hybrids compete a lot.
Yeah, I wonder how off that. I was kind of wondering about like how they set those numbers. Like they could in theory arbitrarily pick 200-to-1 to make their model like look really good for their system. So yeah, like I was wondering about that. If they did some kind of an experiment, like if they released 200-to-1 eggs, how many survive to adults? And then is that comparable to the 10-to-1 ratio? Or is that as if you're releasing 50-to-1? In that case, there's some scenarios where that would help them, right? Because then they can say essentially like you get more bang for your buck if you're releasing eggs. Yeah, but in terms of like this model and this figure, I do kind of wonder if they just like picked that number because it pushed their thing down. I don't know.
I would assume just looking at this is because FS Riddle does the same ratio. It's probably a comparison.
Yeah, of that to the, yeah, and I think that's the most logical sort of like brother kindred technique, if that makes sense.
There's no reason why you can't release adults too, right? They could just raise them to adults and then release.
They could, yeah. And so, yeah, so that would be an interesting control if they would have put in the 10-to-1 adult release. Then how would their model look? That's a good point, yeah. And that since it's a model, they could have easily just popped that in. Maybe they did, yeah. And they maybe, I don't know. No, but so my thoughts are, I mean, I think this is really sort of a revolutionary technology. And I think that regardless of whether what's quote unquote the best, I think the sex sorting with the CRISPR is going to become incredibly useful because there's all kinds of insects that are not sexually dimorphic. And some sort of genetic killing system might be the only way to sex sort things that are not sexually dimorphic. So I think that this technology is really fascinating and really cool. Okay, good?
Let the game real time to die. I shall meet death head on. I shall meet death head on. Decker Kane, you've got to get out of here. Beware foul demons and beasts, there are many foes here. The time has come to cleanse my homeland. I think